The second representative example for understanding of operon is tryptophan operon. This operon controls the biosynthesis of the amino acid tryptophan. This operon is, in short, it is called trypoperon, and which includes usually 5 different structural genes are required for the conversion of the substrate charisma to tryptophan. And usually this operon is an example for a repressible operon. Whereas the discussed one, that means the lac operon, was an inducible operon where the uh, uh, where the operon is in the off stage by default, and the inducer will induce the system, and the process what we discussed as induction. At the same time, in tripoperon, the system by default will be in the on stage. That means there will be the genes expressed regularly, and this system which is already on has to be repressed by a repressor as and when this particular amino acid is not required to be synthesized, and this process is called a repression, right? So that is the basic difference between lacoperon and trypoperon. So these are the two categories of operons usually present in bacterial system. All right. And here the tryptophan molecule act as a repressor. In fact, they act as a co-repressor molecule to activate the inactive repressor protein, and this causes the further repression of the system. And this slide will give you an idea about the structural details of the operon. The 5 structural genes are denoted like E, D, C, B, and A. And there are mRNA molecules. Then there are the genes and different proteins involved in the conversion of the substrate to tryptophan. Okay. And the most interesting part to be noted is the regulatory sites. You can see an operator site and promoter site as we have seen in the lacoperon. Additionally, you can see an additional regulatory site called leader site or TRIP-L-DNA. And within that TRIP-L-DNA, there is an attenuator site. Right. And similarly, upstream to this particular operon, tripoperon, just like the regulatory protein of the lacoperon, here also the regulatory protein is produced by another gene system or another operon system called TRIP-R. And this TRIP-R will produce a protein called a TRIP repressor. And the feature of the TRIP repressor is that this repressor is in the inactive stage, and this alone cannot go and bind to the operator site, and this repressor has to be activated before it has to be bound onto the operator site. And this activation is usually done by interaction with the tryptophan. So when tryptophan is present in the medium, this tryptophan will go and bind with the repressor. Usually this repressor is thus denoted as the aporepressor or in the inactive repressor. So this repressor interact with the co-repressor called a tryptophan and will be converted into an active repressor. And this active repressor will go and bind with the operator site, thus preventing transcription. That will be the condition when tryptophan is available in the medium. But usually, as you know, by default, tryptophan will not be available unless we provide this tryptophan in the medium. So the bacteria has to synthesize tryptophan. So by default, the system is in the on stage. That means there is no tryptophan, and hence the repressor, even it is synthesized, will be in the inactive state and hence cannot bind onto the operator. So the operator is free, which means the polymerase can easily move through, and the structural genes will be synthesized for the synthesis of tryptophan. So that will be the normal condition. And into this, when the concentration of tryptophan increases beyond the limit, or even if the media provides tryptophan, then this tryptophan will activate the repressor protein, and that causes the inhibition of gene expression. So this is the outline of trypoperon. So this is actually the negative regulatory system. So negative regulation occurs in both in lacoperon and trypoperon. Here, this is the representation of trypoperon under negative regulation. Right. All these are about the negative regulation. Now, there is a positive type of regulation, or there is another set of regulation called another regulatory system in tripoperon called attenuation. Attenuation is an additional regulatory system in biosynthetic operons like tripoperon. Tripoperon is a synthetic operon. So, not only in tripoperon, in other operons also you can see attenuation. Right. So, usually you know that once repression is lifted and transcription begins, the rate of transcription is fine tuned or the rate is enhanced by attenuation. Right, in which the transcription is initiated normally but is abruptly halted before the operon genes are transcribed. Okay, so here actually this is an additional regulator that we have to remember. This is an additional regulatory mechanism. So attenuation is regulated by the availability of tryptophan and rely on the close coupling of transcription and translation in bacteria. So that is an important point. We have to remember that bacterial system has a coupled mechanism for transcription and translation. You have to recall the structure in the DNA, that means the trip L region, and this trip L region is going to be mattered in the attenuation system. So this trip L region codes for an mRNA called leader mRNA, and this leader mRNA consists of different sequences, and these different sequences are actually involved in the attenuation system. In the mRNA, this is the leader mRNA. So this leader mRNA consists of 4 different sequences, or 4 different regions are there. It is denoted like 1, 2, 3 and 4. And the importance or the peculiar feature of these sequences is that all these sequences are complementary to each other. They are self-complementary. That means 1 can pair with 2, 2 can pair with 3 and 3 with 4. Okay, so these sequences are very important. And the second thing you have to remember is that within the sequence 1 or within the region 1, you can see 2 tryptophan codon consecutively. There will be 2 tryptophan codons like GGU sequence in the sequence 1 or in the region 1 of the leader mRNA. And usually these, since already stated that these sequences are complementary to each other, the sequence 1 can pair with 2, 2 can pair with 3, or 3 with 4. So once 2 is already paired with 1, then 2 cannot form a pair with cannot form a structure with 3. Or if 3 is already paired with 4, then 2, 3 will not be formed. So that will be the situation. So once 1 bound with 2, then there will not be any 2, 3 structure. Or similarly, then the remaining possibility is that 3 can pair with 4. But if 2 pair with 3, then there will not be 1, 2 structure and 3, 4 structure. Okay. So usually the complementary pairing of sequences 3 and 4 is denoted as attenuator. Remember, this pairing of the structures 3 and 4. When the region 3 and region 4 pair each other, that structure is called attenuator. Usually these self-pairing causes formation of a stem loop structure, and this stem loop structure is what we call the attenuator sequences, so attenuator structure. Usually the sequences base pair to form a GC rich stem loop structure, and we know that when you closely analyze this leader mRNA or the leader region in the DNA, just after this sequence 4 there is a uracil rich region in the mRNA. That means this U will be pairing with adenine in the DNA. That means there will be UA pairing, and which is already we know that it is a very comparatively very weak bonding. Okay, so the formation of stem loop structure adjacent to the U rich region causes the mRNA to dissociate from the transcription complex. So there will be a termination process. And this will give you a detailed broad explanation for attenuation. There will be 2 conditions. In the first situation, tryptophan is available. Tryptophan is abundantly available in the system, in the cytoplasm. Then in that case, when the trip mRNA or the leader mRNA is synthesized, normally transcription is followed by translation in bacteria. So the translation will be following. That means the ribosome will bind onto the uh, leader mRNA, and this leader mRNA will be translated by the ribosome. And for translation, we know the mechanism. The ribosomes has to be supplied by the amino acid, and this is done by the tRNA molecule. And this will continue, the synthesis will continue, and the ribosome will reach the trip codon, which is present in the, remember, you have to remember the point, the trip codons are present in the sequence 1. There are 2 trip codons in the sequence 1. So the sequence 1, when the ribosome reaches that trip codon, since tryptophan is available in the system, the trip tRNA will, or the tRNA will supply tryptophan into the ribosome, so that ribosome can move without any delay, without any complication, and the ribosome will pass the sequence 1 and will reach into sequence 2. Okay, so meanwhile the RNA polymerase, the RNA polymerase would have moved 3 and 4. That means there will be synthesis of 3 and 4 completed by that time. Okay, so when two synthesized, 3 was beginning to, or just starting to synthesize, but when 3 is completed, 2 has been bound by the ribosome. That means 2 cannot form a stem loop with the 3. Okay, but when 3 is completed and RNA polymerase is moving further, the 4 is also synthesized, and 3 or 4 are hence they are complementary to each other, they can form a stem loop. So this stem loop formation causes dissociation. This is like, when there is a pulling of this mRNA molecule from the formed by the, or this pulling is effected by the stem loop formation, and this causes dissociation of the entire complex, and there will be termination. That means when tryptophan is present, we do not want to synthesize the enzymes required for tryptophan synthesis. That's why there is a termination. Then in the next situation, the other situation is that when tryptophan is absent, there is no tryptophan. What will happen? The ribosome will move through the sequence 1, and when it reaches to the trip codon, that means when it reaches the GGU GGU codon, then what happens? The ribosome will wait for the tRNA, and the tRNA has to supply the tryptophan. But the tRNA cannot do that because there is no tryptophan available in the cytoplasm. So even the ribosome wait for a long time, tRNA cannot supply. So the ribosome will be halted. Ribosome will be halted in the sequence 1. So the sequence 1 is covered by the ribosome. So at the meanwhile, what will happen? The sequence 2 and 3 would have synthesized, right? And 2 and 3 are, since they are self-complementary, that means since they are complementary to each other, 2 can pair with the 3, and this causes a stem loop, 2-3 stem loop. And this 2-3 stem loop is otherwise called anti-attenuator or anti-attenuation stem loop. And 4 will be free. 4 is not available, or 3 is not available to form a stem loop with 4, right? Since already 3 has been bound by 2. So 4 is free. So this causes the RNA polymerase to continue transcription through the structural genes, right? That means there is an anti-termination. There is no termination of transcription due to the ribosome stalled in the sequence 1. So this is about attenuation. Hope you can follow this. So these are the some points to remember for better understanding of attenuation. That is, transcription and translation are coupled in bacteria. Then there are 2 consecutive tryptophan codons in the sequence 1 of the leader mRNA, and tRNA carrying tryptophan is essential for translation to proceed beyond the trip codon in the mRNA. So these are the points you have to remember for attenuation system, and I hope you follow this discussion. Thank you.